Welcome back. This part of the talk will give you a very basic overview of what our immune system includes and how it can react to microbes. Going into details about all of this can take hours, so we've only scratched the surface of what's important for your basic understanding and try to make sense of the mechanisms of immunity. First of all, I want to make sure you're familiar with a few terms we'll be using a lot beyond this point. Antigen. Antigen is any substance that can induce an immune response. And given how our immune system works, this can pretty much be any kind of substance. Most of bacterial and viral antigens are proteins, so it's generally okay to think of antigens as proteins, at least for the purposes of this guide. You will probably hear the terms antigen and toxin used interchangeably, as any bacterial protein or toxin is an antigen. Mind that high molecular mass substances have so-called antigen sites. It means that specific parts of the foreign substance can cause immune responses. What this implies is that for any given toxin, we can potentially identify hundreds of suitable sites where our antibodies will attack. Receptor. In biochemistry, it's a protein structure that can bind some substance and induce a reaction, generally used for signaling and responding. For example, our cells change their metabolism when certain hormones bind to their respective receptors. Immunity is the ability to resist infection. It can be innate, aka the one we have from birth. You can refer to my previous example of HIV immunity due to specific gene mutations. Another example of this is how many of the viruses that affect animals don't affect humans and vice versa. We simply have no target cells for those viruses to attack. The second type is adaptive, aka the immunity we develop throughout our life. It can be natural, which is divided into active, aka when you contact the disease, or passive, through receiving maternal antibodies from breastfeeding. And it also can be artificial, through immunization, or what we'll be discussing in here, also known as vaccination, or passive by transferring antibodies from using serums for medical indications. Immunity can also be classified as specific and non-specific. Specific immunity only affects one exact pathogen. For instance, you get the flu and then the next year you get it again. Your immune system did develop immunity the first time, but now there is a different strain of the virus that your immune system can no longer recognize. You developed specific immunity against the first strain, but it didn't cover the new virus. Non-specific is the opposite. By suffering a disease, your immunity covers a larger variety of strains of the same virus or bacteria. If you've been immunized against hepatitis B, you will be immune to at least 90 to 95% of circulating hepatitis B strains. Non-specific immunity also includes mechanisms of immunity that can target a variety of pathogens with little in common. Interferons will inhibit the growth of most viruses, and they are not specific. NK cells, short for natural killers, have a variety of receptors able to target many alien cells. By mechanism of action, immunity can also be divided into humoral and cell-mediated, which will be covered using an example for better comprehension. In immunology, the immune system is viewed as consisting of multiple components: organs, cells, and molecules. We'll start with cells. There is a myriad of them that regulate how our immune system responds. For simplicity, let's divide them into these categories. First, granulocytes. Leukocytes, aka white blood cells, can be divided into ones that have granules and ones that do not. Granulocytes, the ones that do have granules, are mainly responsible for inflammation and non-specific immune response. Lymphocytes. Lymphocytes are a type of leukocyte. These cells have a variety of classes and functions and essentially take care of the entire immune response, going from non-specific during the onset of infection to specific later on in the course of the disease. These include T cells and B cells. B cells are the simpler ones to talk about. They make antibodies which bind to antigens. This type of response is the one we call humoral. Humor means water in Greek, which in this case refers to blood, as antibodies circulate in our bloodstream. T cells are more complex and regulate pretty much everything about our specific immune response. These are divided into T helpers and T killers. T killers are T cells that are trained to find and destroy a specific pathogen. Think of how the Terminator from the movies works. They have a target and they're out to get it. T helpers help other cells grow by presenting antigens and using specific molecules to control a cell's growth. They're responsible for making both other T cells as well as B cells, as well as controlling the intensity of our immune response. They are often divided into type 1 and type 2 helpers, responsible for T cell and B cell growth respectively. Their balance determines which type of immunity is stronger in a given individual, and also what types of problems with immunity a person is likely to encounter. If there are more helpers responsible for B cell growth, there is a higher chance of typical antibody-mediated allergy responses such as pollinosis and anaphylaxis. If it's the other way around, there is a high chance of contact dermatitis. Antigen-presenting cells. As the name implies, these cells are equipped with a variety of tools to kill microbes, split their antigens, and use those pieces to present them to growing lymphocytes. These cells, in a way, help to identify the pathogen and create lymphocytes that will be able to tackle it. Kupffer cells can be used as an example here. A huge amount of those cells is situated in the liver. Patients that present with acute liver failure have a much higher chance of dying due to infection. Their immune systems enter a paralyzed state if they are unable to properly identify the pathogen and react accordingly. Organs are generally divided into primary and secondary. Primary organs include bone marrow and thymus. Bone marrow is where our immune system cells are made, all of them. Without bone marrow, there is no immunity at all. Stem cells have been a popular topic among people in the early 2000s and even 2010s. Bone marrow is pretty much the only source that can still provide them in large enough quantities. Thanks to stem cells, bone marrow is able to produce whatever types of cells it needs to, and in large quantities as well. Thymus is a place where T cell precursors move to grow into fully fledged T cells. While they are made in bone marrow, their primary place for growth is inside the thymus. Thymus is an interesting organ as it exists up until a certain age. Generally, it gets fully replaced with fat by the age of 25, and T cells create a place for themselves in other organs. No one knows exactly why this happens, and many believe it to be one of the primary reasons we age in general, and also why our immune system weakens later on in life. Secondary organs include spleen, lymph nodes, and other sources of lymphoid tissue, such as tonsils and mold. In these organs, cells present antigens to T and B cells so that specific immunity can develop. Essentially, once lymphocyte precursors leave bone marrow, they also find a place in secondary organs where they develop and grow independently. Immune system's molecules include substances that mediate the immune response through and through. There is no point in discussing interleukins and other types of transmitters as they won't enhance your understanding of vaccination. If you want to, you can do some reading on your own regarding those. A distinct non-specific defense mechanism against pathogens is the complement system, which you can look up if you also want to. What we'll focus on is specifically antibodies. Antibodies are a class of plasma proteins produced by plasmacytes, the fully grown forms of B cells. A typical antibody has 3 active centers, 2 antigen binding centers, and 1 so-called cellular. Immune system cells have receptors for cellular sites, so if an antibody binds to a microbe, it can attract other cells to come finish the job. When it comes to antibodies, there are numerous ways they can neutralize threats. Some specific examples will follow because the main idea behind vaccination is indeed forming antibodies against certain antigens. There are 5 classes of antibodies. For the sake of basic understanding, I'll briefly mention 2 of them. Immunoglobulins class M. These antibodies are the default mode of B cells, meaning a B cell will produce them unless it's commanded otherwise. They are formed at very early stages of an infection. They look like this, have 10 antigen binding sites, and are not specific for this antigen. Essentially, it's a let's throw something at them and see what works strategy, which, to be fair, is a very valid strategy as these antibodies help facilitate further immune response. Immunoglobulin class G. These are the antibodies you're already familiar with. They form at later stages of an immune response once B cells were exposed to an antigen and had some time to grow. They're highly specific for a particular type of infection. There's a lot more of them and they remain for a long amount of time. These were a few concepts you needed to be familiar with, and next up we'll look at an example model of an immune response.